Yes.
Here we are.
Larvae.
Larvae. Possibly the cutest little baby pictures you'll ever see.
Oh, adorable, adorable.
Yeah. So this chapter actually came out. It was sort of it came in the second tier. Once I started realizing that we did have quite a number of larvae that maybe we had enough for a chapter, and then it just blossomed into this global search for sunfish larvae. And so I think we have organized this talk into 2 parts. I'm taking the first part, right?
Yes.
And then I will hand off to you, and you will and you'll pick up. Let's see. Yep, I'm going to hand off to you right after we talk about the ways of imaging larvae, and then I'll hand off to you. Okay.
Yep.
Alrighty. Well, so I will. I'll be sharing my screen now, and I'll see you, Mayana, in a little bit.
Yeah.
Okay. Let's see. Share my screen. Share. And here we go. All right. Is that working? Let's see. Yep, that's working. Okay. All right. Well, as you'll note, with all these talks.
Sorry, Tierney, the screen share didn't start.
Oh, it didn't.
Nope.
Oh, all right. I'm glad you mentioned that. There we go.
There we go. I see it now.
Okay. All right. Is that working now? Yeah.
Oh, yeah.
Okay. All righty. So as you'll hear echoed throughout this whole symposium, this has been such a collaborative effort. And this chapter, we have lots of people to thank who were involved in helping look for larvae, putting in their sightings and sharing their reports. So a big shout out to everyone on this slide. John, Inga, Toshiyuki, Marco, Leah, Rachel, Katsumi, Gento, Tor, and Lucas. Thank you very much. And I actually want to show you, we put out this call to everyone we knew, every fish collection, whoever had fish larvae, if they had any sunfish larvae. And then bit by bit, we started to put it into a big chart. Mayana started this big chart, and we started to fit them in. And this not only included modern day fish collections, but plumbing the literature back hundreds of years and trying to make sense of all these little drawings that have been made of the Molidae, dating back many, many years, and trying to place them into the proper genus and species. So this was really detective work at its best, and so much fun because they're just some of the cutest little larvae imaginable. And I'm just going to show you, as we went and found different larvae, that this was a pretty Herculean effort. Lots and lots to wade through. A big shout out to Don Kors at the Stanford Library, who was invaluable in finding esoteric, ancient literature sources. So this is just a snapshot of some of the resources and some of the little drawings that we sorted through to try and figure out who was whom and what ontogenetic stage they were at. So as you can see, it's a lot of work, some based on misidentifications that we were then able to try and place. And once that was done, and albeit with a lot of help from people mentioned at the beginning of the talk, we also tried to map where those use the metadata from those reports and map them across the globe. And here you can see this map. Thanks to John Ryan for helping put this together. And we have a lot of sightings of Ranzania. If we looked at our total record of of larvae that were collected, dating all the way back to the 1800s. We had about 452 records, which comprised about 9,770 larvae. So 285 of those were Masturus, 84 were unspecified Mola from the Mola genus, 61 unspecified, just the Molidae family, and the bulk, by far the bulk of the larvae were Ranzania, so 9,340, which with about 340 eggs. So now inherent in this search is the greater search of trying to find the spawning areas of the Molidae, of any of the species. And you know, you'll if you stay tuned in, you'll be able to listen to our reproduction chapter that's being headed by Christy Forsgren, Richard McBride, myself, and Toshinaka Tsubo-san. That Mola are incredibly fecund. They have they produce an enormous amount of ova, millions, hundreds of millions in a single individual. So it's amazing that there aren't just more examples of the larvae. And then also, we have never, ever seen them spawn in the wild. So that's another big mystery. But we do see areas where we tend to find them. So the Sargasso Sea is a large one, and we'll be having Marco Fries will talk specifically about his work in the Sargasso Sea and with Ranzania larvae. We also have hot spots off of Hawaii. John Whitney has certainly collected a bunch out there. And Marianne has gathered many, many larvae off of Australia. So we're bit by bit putting together the global map of where we find eggs and larvae, and hopefully be zeroing in on where the spawning areas are of the sunfish. Now, ways of imaging larvae, you know, that's one of the key parts, is trying to identify which larvae goes to which genus, and that's been tricky. But we have some amazing ways of imaging larvae and looking for those specific defining features, and Marianne will be going into what those features are that designate each larval form, in each genus. But here are just some examples of some beautiful photomicroscopy work done. This is Ranzania up in the upper left corner. Frank Bainch out in Hawaii has raised some Ranzania from eggs, which is really remarkable. And I love these images of Dave Johnson and Wittenrick of additional little Mola larvae. Recently, we have some micro CT images that are just spectacular, taken by Kate Bemis, who will be giving an entire lecture, a presentation on the anatomy of the sunfishes. And really brilliant work there. I think these guys really look like something out of steampunk. And a recent paper by Shono et al. That shows cleared and stained sunfishes. So we have some really good imaging techniques that can hone us in on those details to help us identify who's whom. And now I think I'm gonna stop sharing. Let's see if I can do that. Oh, it seems like my screen. Okay, stop sharing. Thank you, Joe. My screen stopped there for a second. And I'm going to hand it over to Marianne to take the ball and run with it.
Excellent. Hi again, everyone. So I will be talking about the Molacanthus conundrum now, and that has to do with the early life stages of the Molidae. And it's easiest to start with a more normal fish and look at how the ontogeny is there, or the early ontogeny is there. And this is a diagram from Kendall, and it shows the early ontogeny of jack mackerel. And this is going to be incredibly brief and superficial, but just so we have an idea of what's going on. But basically, we have eggs with an embryo that develops inside. It hatches to a larvae with a yolk sac that then is absorbed. And as the larvae develops, it starts to develop the fin rays And it's hatched with a notochord and a fin fold, and this basically becomes the tail of the fish. Once it has all its fin ray complements, it's usually considered that it transforms into a juvenile. So this stage here is never clear in fishes, or seldom clear, and sunfish are not the only taxa that fits in poorly with this, but perhaps sunfish are an extreme example of how they just do not fit into this at all, where the transformation from larvae to juvenile is when the full fin ray complement is in place. It has lost any spines it had during the larval stages, and the proportions of the body are similar to the adult. So for Molidae, they hatch quite late, or they're quite well developed, advanced in development when they hatch. So they actually don't really have a proper yolk stage up here. So they hatch more like this. They then go through and they immediately do something else than the rest of the fish, and they go through what's called the ostracian stage, the molacanthus stage, before they transform into juveniles. And the Japanese Broadcasting Corporation has made this brilliant little animation of how this happens. So that's the molacanthus stage there. And then it transforms into a sunfish. And I want to watch it again because it's so good. So this is the little larvae that hatches with a notochord, transforms into the molacanthus stage, and then into the sunfish. Right, so this diagram here is from the book, and Tierney and I spend a lot of time trying to make these growth series for the three species, Masturus, Mola, and Ranzania laevi. So these are basically they're modeled on published drawings, and we try to kind of represent the different looks in the different at the different sizes. And if we start up here, we don't really know what goes on in Masturus lanceolatus at this very early stage, but both Mola and Mola Ranzania laevis hatch with a notochord and a fin fold, just like normal fish do. And for this reason, this stage is often referred to as the tetrodont stage. And then immediately something else happens. They start to lose this notochord. It atrophies simply. And as the dorsal and anal fins start to develop, it atrophies here. And they turn into these little spiky balls. And that's sometimes referred to as the ostracian hoop stage. Transformation in terms of the full fin ray complement happens around 4 or 5 millimeters long. And so really, according to the tradition, they're no longer larvae. But they're not juveniles either, because they do not have the normal adult body proportions at all, and they still have all of these really funky spines. So this in-between stage, before they have lost all of their spines and they have the general proportions of the adult, is termed the molacanthus stage. And it's sort of an in-between stage that's just quite extraordinary for this group of fishes. Interestingly, Ranzania does not go through that. So they basically transition from a little spiky ball into something that starts to look much like the adult in terms of body proportions. So these guys during this stage are taller than they're long. So they have the wrong body proportions, and as they grow, they start to elongate and round out a little bit. Up here in the corner is Mola alexandrini, and this is from some work we've done down here in this neck of the woods in Australia and New Zealand, where we have a really, really neat collection of specimens that has been collected by the Australian Museum since 1925. They've collected these larvae. They have over 30 of them, and we have a really good growth series. So I'm working on describing them, and we were lucky enough to be able to genetically verify that this is Mola alexandrini. So in terms of where do these ostracian and molacanthus stages, where do the names come from? And it's a little bit neat because it goes back to and touches upon the history, this crazy taxonomic history of the sunfishes, because back in the days, and we're back in the 1800s now, all the way back to the 1700s, the early naturalists, when they saw these little early life stages, couldn't imagine that they would grow big to the big sunfishes, so they described them as separate species. And this one was described as Ostracian boobs, but we can now see that it's an early life stage of Ranzania labies. The Molacanthus stage comes from, was coined by Swainson. He modeled his species Molacanthus pallasii on another species that was described by Pallas in 1770 called Diodon mola. We can now see this is an early life stage, or a Molacanthus stage mola. So that's where that name has come from. So the interesting thing is, what's the function of this stage? Why do they go through this remarkable transformation? And we don't really know yet, and this is a really interesting area of research. But it could be because the sunfishes need to conquer an exceptional growth spectrum, and perhaps this is what they need to do to climb up the size ladder. And at some stage, they need to look like this. So the next thing we did was we also wanted to know if these early life stages are eaten by anyone. And so we did a very large literature search, although it isn't exhaustive. And we found lots of examples of predators that had eaten early life stage Molidae. And all of these records were really difficult to standardize and summarize in any way. So there is basically three different types of observational groupings here. There's like discrete observations where we have a fish that has, there's Molidae has been described from its stomach, but we don't know anything else about what it had eaten. And then we have diet studies where a predator's diet has been studied, and we can see Molidae as part of that. And then we have narratives where it's mentioned that this or that fish eats early life stage Molidae. But some of the records were more believable than others. And here we have Bieber from 1934 describing the deep sea and how this saber-toothed viper fish is eating all these mola larvae that we can clearly see are Ranzania. So we weren't quite sure what to make of this observation. So we're a little hesitant to include the viperfish in the list of predators, but who knows? So here they are. So there's a lot of different species of fish, but a commonality is that they are pelagic. So these are species you would catch on pelagic longlines. We also have a range of seabirds, especially this one here, the red red-tailed tropic bird, I think it's called, that in particular eats Ranzania larvae. Interestingly, though, when we look at all of this, most of the records are Ranzania, so they seem to be eaten more than the others, or found more often. But we also have a lot of unidentified, just called Molidae, so who knows what's in there. So there is still a lot to learn here, but one thing that I think is striking is that given how fecund these fish are, there must be so many larvae, and once they reach the Molacanthus stage, they would seem a good little morsel to eat. So why don't we see them more often in diet studies? So that is a real good enigma right there. Right, that's it for me here. Tierney, please come back for questions.
Yeah, so that was great, Marianne. Yeah, so many interesting questions that arise from this. Like, what is going on with that little Molacanthus stage? And is it related to their? They do have a swim bladder when they're little, and then they lose their swim bladder, and but then I don't know if anyone's seen if Ranzania have swim bladders when they're little larvae as well.
I don't know either, but this would be a really interesting thing to look at, because Ranzania is a very different fish. So that to me is almost like a standard bony fish.
Mm-hmm.
they don't exhibit a lot of the traits, and they don't have to climb this massive size spectrum either. And so it's interesting that they don't have that Molacanthus stage. And it would be very interesting to see whether they have swim bladders.
Yeah. Although Giorgio this morning was saying that in the fossil record you have Ranzania that got to be very large, like 2 meters. So that's sort of interesting.
Yes.
But certainly modern day don't get over 80 centimeters. So yeah, no, I mean so many interesting questions.
Yes.
And I really look forward to seeing the ontogenetic sequence of your Mola alexandrini to add to the lot. And then someday Molitecta, but no one's ever seen a larvae of Molitecta. No idea what that looks like.
And of course, I'm very keen to look for them.
Yeah.
So we need a lot of funding, Tierney. So you need to come down here. come look for larval Molitecta with me. But I can see we have a really good question here. How long does it take for sunfish to fully mature? And I think this is an interesting question, because I think maturity is reached around 1.5 meters. I think the latest research is showing, but we don't know exactly how long it takes to grow to that stage, because growth rates in the wild are unknown. So based on sunfish in captivity, it doesn't take very long at all to reach 1.5 meters.
Right. I mean, you can put on 800 kilograms in 14 months.
Yeah. So the potential for growth is enormous in these fish.
400 kilograms
to the wild is another story. And I mean, we have got some from the photo identification work that we're doing in Bali. We have the same fish over time, and I don't think they're growing that fast.
Yeah.
So there is, So the answer to the question is that we really don't have a good idea of how long it takes in the wild.
But they can reach the full size, we're thinking, sexual maturity probably like 3 to 4 years. You don't see anything reaching sexual maturity like under 1 meter,
In terms of the sunfish?
Yeah.
Oh, yeah, and I think, there has been some work done, and I think it's sort of around the 1.5 meter mark.
Yeah.
And I guess in captivity, a sunfish can reach that, like you say, in 3 to 4 years. But whether that translates to the wild is what.
Wild. Right.
But I don't know how fast it can happen in the wild. I mean, they work pretty hard for their food, I think, in the wild, and in captivity, they don't really have to do any foraging, or they don't have to. Food is easy to come by in the aquarium.
And it's more difficult to age them using otoliths. Like you can use otoliths, the little ear bones that we use to age salmon and other commercial fishes. But with Mola, it's really difficult to age them using their otoliths. It's not really possible.
Yeah.
They accrete in a different way. They're made out of vaterite, and so it's still, how to age Mola is still an ongoing challenge, So, yeah, but great question. Keep those questions coming in, and we will try to answer them. Mayana, you've made your email available to field additional questions. Mine is tierneytees@gmail.com. So definitely send your questions to us, and we will just keep continuing on